Hormones, hormones, hormones here and there. Hormones, 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 hormones everywhere. Hormones, 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 hormones everywhere. Yeah, the hormones from our thyroid for our metabolic rate. And gonadotropins, they make women ovulate. There's hormones in the kidney and there's hormones in the brain. Hormones help you sleep at night and help me stand the pain. Hormones from the pancreas to control metabolites, and leptin from the fat to control our appetites. There's feedback loops and circuits that are beautiful to watch, and in this course we'll study them a lot, but not too much. Hormones, hormones, hormones here and there. Hormones, 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 hormones everywhere. Hormones, 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 hormones everywhere. Welcome back to Hormone Circuits, Lecture 2. I loved talking with you about weight and leptin so much that I decided to do a part 2, so that we can go deeper. Lecture 2 on weight. And the purpose in this lecture will be to, first of all, take a nice deep sigh of relief. So I invite you together with me to take a nice deep sigh of relief. Is to get an insight in how we work, because this weight control project is in fact part of our research. It's not published. This is a world premiere, and you're the first ones who get to see it, how we work with the experimental data in order to understand models like this of complex systems. And also to return to the question we had last time, what happens if you eat food that's intrinsically more fattening? What happens, you gain weight, but do you eat more grams per day or less grams per day? And questions like this. What happens to us when we age with our weight? And what happens when we exercise and things like that? Those will be the topics of today's lecture. So I'm gonna, as always, write down for you where we are. We did welcome, breath. Now I'd like to review what we did last time, so we have a basis for today's lecture. To give you space, breathe. So we consider the question, why is it that our weight is nearly constant over decades? Give or take 5 kilos, it's about the same. Of course, there are times where we fluctuate and deviate, but overall, it seems there's a set point for that, our weight, and where that set point is has been mysterious. And research over the last century pinpointed several hormones. Chief among them is leptin, that helps us when we have more fat to eat less, to control our appetite. And so we discussed a feedback loop, which is fundamentally food intake makes us fatter. Fat produces leptin. Leptin shuts off our food intake. And I just want to mention that leptin is produced by fat, but only when you eat. So food also affects leptin production, and that's a great evolutionary mechanism. So even if you have some fat, but now you're starving, there's no food, the body prevents the formation of leptin so that you still eat. It's a kind of important part of the design here. And it's important, as we said, to keep weight under control, because if you have too little weight, too little fat, you're very prone to starving. So basically, we can live off our fat for a few weeks. Of course, we also need to break down proteins to make glucose for the brain and things like that. And if we have very little fat, we can survive less time without food. And also, without fat, women can't become pregnant or ovulate. So fat, and also mice, is very important for reproduction and for reaching puberty. So it's good to have not too low fat. On the other hand, it's good not to have too much fat, because then you can't escape predation, and you're especially tasty to those owls that eat the rodents, etc. And so there's an evolutionary set point, and it makes sense to have fat at a sweet spot. So we discussed this feedback loop graphically using the concept of the phase portrait, where we plot our variables, U, let's say, in grams per day, and percent fat. Okay? And we discussed two kinds of experiments that can help us make sense of the set point. The first kind of experiment we call the diet. We made the diet line. So suppose you force a certain amount of grams per day of food, and you wait for a few weeks and see where fat percent is stabilized. That is to say, the steady state fat percent for a given amount of food. And that makes the diet line, which we propose as a straight line with an intersect at a certain food level. Below this food level, you die. You have actually zero fat. And that's the diet line. And so we're going to see today, is it really an experiment? Is it really straight? What is this U0? Questions like that. And then there's the appetite line. The appetite line is a different kind of experiment. It's after you make a diet and you reach a certain fat, let's say high fat percentage, how much will you eat during the next 24 hours? What is your appetite? So we have a lot of fat, the leptin production, we have a lot of fat and you've eaten a lot. By the way, this arrow here is not instantaneous food. It's how it integrates over food in the past few days in a way that's a little bit mysterious. It has to do with insulin. It's not exactly clear how this works. But when you're fat, you eat less. So the appetite line looks like this. And you can measure it, as I said, experimentally. And so notice it also intersects the x-axis. This is the satiety point. Let's call it U satiety, which is, even if you have zero fat, you can't eat infinite amount of food. There's some point where your stomach will get so distended and will produce hormones like GLP-1 that are satiety hormones, and food is no longer rewarded. So there's an intersect point here. So we talked about these two all kinds, and the important point is where they intersect. Because when they intersect is when your fat matches the amount of you eat produces the amount of fat that gives you exactly the appetite for the amount you eat. So it's when you're fat and your appetite, diet and your appetite are matched, and everything is matched. And we also talked about dynamics. So what happens if you start somewhere? The first, so a certain amount of fat and a certain amount of eating, let's say force you to eat this amount, you still haven't gotten to maybe to the steady state, it doesn't matter. Now we'll let you eat freely. Eat freely is called ad libitum in Latin. Eat as often and as much as you want. You'll shoot to the appetite line. So, and then you eat a lot, more than your set point, more than you usually do. And then you get fatter, and then you eat a little bit less, fatter, you eat a little bit less, fatter, you eat a little less, until you come back to the fixed point. So fixed point. So this phase portrait actually shows you, no matter where you start, if you start here, you're going to undershoot, eat less than you normally do, and then fatten up in this particular way. That's the theory. So you see the dynamics and the fixed weight. The weight set point, according to this theory, is encoded by two lines. One is the energetics, food goes to fat, and the other one is appetite. Fat goes to leptin to reduce food intake. And the balance of those two processes determines your weight set. That's the important point of yesterday's lecture. Not yesterday, last week's lecture, lecture 1. Let's take a nice deep sigh of relief. And now we would like to compare this to experiments. And I just want to highlight Alon Bar. He's one of the two teaching associates in this course, Avi Mayo and Alon Bar. You're welcome to turn to Alon and Avi with any question about the course, exercises, etc. And Alon's PhD project is this right now. And Alon did projects on hormone seasonality that we'll hear about too. What I'm talking about here is research going on in my group. You can't find it in any paper so far. It wasn't published. So you're the first to see this explanation of the weight set point and all the dynamics. It's a world premiere. So congratulations to all of us. And I guess in this lecture, I want to expose a little bit how Alon works. How do we do research in our group? which is it takes use of about a century of amazing experiments that people have done over the years with different kind of questions, and it gives detailed data that you can reinterpret in test theories. This. So we're going to do that right now. And in particular, Alon found very interesting the work of Harris and Martin from 1986, University of Georgia, whose idea was to see what happens to the body composition, fat and protein, etc., of mice that are overfed and underfed. And they conducted one of the best experiments, detailed experiments on the rat model organism. That's a model organism often used to study metabolism and nutrition. And in their paper, there's data that's, when you transform it in the right way, it can actually give you these null points, and we can test whether they actually look like this, and also test the equations that we're going to write down and see what the parameters are and how the dynamics look, and understand quite deeply what is going on with feeding in this model organism. So we're going to discuss the data of Harris, Martin, and what Alon did with it. In this paper, Harris and Martin looked at three conditions. One is when the rat is fed normal amount of food. Another when it's underfed, so it's fed 40% of its normal diet. That's really very little, either by just giving it restricted diet or by tube feeding, so you're sure how much went into its stomach. And another situation of overfeeding, 160% of the normal diet, really stuffing the rat with tube feeding. So these experiments are, of course, unpleasant, and I apologize for that. Hope you're not triggered. And then after a few weeks, letting the rats reach a steady state weight, stopped this forced feeding and let them eat ad libitum, as freely, as often, as much as they want, and measured food intake every day for a few weeks until they kind of reached a new steady state. And that gives us the perfect experiments for the diet line and the appetite line. So let's see how the data looks like. And so I'm going to plot here time in days. So this is food intake in grams per day. And these rats eat 15 grams per day. That's their normal ad libitum rate. And in this span of time here, let's say these 30 days here, food intake is now raised to 160%. And I'm going to plot here now as a function of time the fat in these mice that start from about 35 grams of fat, can experimentally determine. And during this time of overfeeding, the mice gained weight, gained fat. It looks like this. And they start to approach a steady state. Now, so this is the overfeeding period. Overfeeding is stopped, and you have the recovery period where the rats can eat whatever they want as often as they want. And what happened is that they ate less than 15 grams. Gradually recovered. And over this time, their fat dropped. Recover. So this is the overfeeding experiment. I'm going to draw here in a different color what happened when there was underfeeding. So in underfeeding, food was dropped to 40% of its control value in the natural eating rats, and weight dropped, fat dropped. In fact, you couldn't do this experiment much more because the fat dropped almost to zero. Once fat drops, the body breaks down muscle, and it's considered lethal to have rodents lose more than 50% of their weight because they start to harm muscles in their heart, etc. You stop basically animal experiments a lot of times when they reach 50% of their weight. And then after this, so this is underfeeding, they overshot. They ate more than they usually do and recovered. And their weight went back. Yeah. And I'm going to ask you now to ask me a question by chat. The people have already asked me some questions. Okay, so the first question, during these first 30 days, fat is increasing. There's no response of food intake level. That's the question. The answer is they forced the food intake level by tube feeding. So the mice couldn't do anything about their food intake level. That's the gruesome part of the overfeeding and underfeeding. They're forced. I hope that answered the question. How can we create persistent changes to appetite line? So how can we change our appetite line and become skinnier? So that's towards the end of our 90 minutes together. We'll get to that. To that burning question. During the controlled part, the researcher controlled the amount of food intake. Alon answered. So see, Alon not only did this work, he's also answering your question by chat. So check out. Mice gain weight over time in our animal houses. How did you prevent this? Okay, so Dan says, in our animal houses here in our university, for example, rats and mice, they gain weight naturally with age. Right? So what I'm plotting here is fat divided by control. So and then I'm going to change this here to 1. So that means even though the weeks move and the mice gain weight, this is always divided by the control weight. I hope I answered your question, because rats keep growing weight over time, and that's something to be controlled for. Ida, does that make sense to you? Say yes or no in the chat. Are these lines mirrored? Is a question. They're not exactly mirrored, and I'll show you the numbers exactly, but they're qualitatively mirrored, right? They're qualitatively mirrored, but the numbers are very important for us. So thanks for these questions. And now we notice that these experiments just give us points on these lines. And why is that? Let's first take a nice deep sigh of relief as we move from this time data to this phase portrait data. So we said experiments, time, and now we're going to the phase portrait data. Why is that? Because why is that? Because during this controlled feeding, we can look at the steady state fat. So I can now put some numbers on this UF plane. First thing I'm going to do, UF. So I'm going to put these experiments in these UF plane. First thing I'm going to do is I'm going to rescale things so that the control food intake is 1, and the control fat percentage is 1. So that means the control mice eat 1 and have fat 1. So I'm just changing the units here. Now, the overfeeding experiment, or let's start with the underfeeding experiment. The underfeeding experiment forces you to be 0.4, 40%. That's forcing. Since it's forcing, we're talking about the diet line. The steady state fat turns out to be nearly zero. Okay, so we have 1 point on the diet line. The overfeeding experiment is 160 times percent the normal feeding, 1.6 in this dimensionless units, food intake. And the fat at the end of it is a little bit above 2, twice. And nicely, just a little bit above the line, but it didn't reach steady state yet, this one, it's clear, but that's the data point. Nicely, these points are not too far off this line. And by the way, this line is very constrained. If I say it's 0 at 0.4, it has to move through 1, 1. That basically determines this line. So this is experimental prediction. You can say that is reasonable because this is still didn't reach steady state. And okay. So that's the diet line. Diet line. And now we have some parameters on it. Like we know what this U0 is. This minimal food for zero fat. And now we can also look at the appetite line. What's the appetite line? Appetite line is defined as following. After I let the animal eat freely and it has a certain amount of fat, how much will it eat? So in this situation, the starving rats, 0.4, 40%, they went up, right? And they, these rats have very little fat. They have very little fat. And they ate here about 50% more than, so this point here, let's make it a triangle, is here. And then if I follow them day by day, I say, okay, now they gained a little bit of weight. I know their weight, and then in this day, and how much they ate. I know the weight and how much they ate, because this is the free eating line. They basically do this. And the other experiment where overfeeding, and then the overfeeding, so they are fatter. There's this amount of fat here. What happened to them? I'm going to draw these. Let's draw these as circles here. What happens to them is, first of all, notice what's happening. Their food stays constant. Their eating stays constant, while their fat rapidly recovers. Fat rapidly recovers. Their fat goes down, but their food intake stays about the same. So it looks like this, and then they start converging. And this gives you a shape that looks like this. And so this experiment fits very nicely with the qualitative model that we're going to write down based on the biology of this feedback loop. It has a kind of linear diet line, a linear looking appetite line that then rises very quickly. It's as if in this situation here, when you have a lot of fat, you basically stop eating if you can. You stop eating in order to drop fat really quickly. It's like you devote everything in order to lose fat when you're overweight, showing maybe how important it is not to have too much fat in the natural world. It's a very fast response, and then it's like a bang bang. You devote everything to losing weight in this particular part. All right. Actually, the theory, as I'll show you, will also give us very steep. We can't see that in the experiments, but the theory is very steep. Also, when you're under, you have too little fat. You eat a lot more in order to go in this in the theory in order to go up in this direction. I'll show you that later. So there's like in the extremes, there's an emergency situation. And let's take a nice deep sigh of relief. And so more questions. The question is, do overfed mice catch up to the control mice weight? The overfed mice actually, in the end of this experiment, are heavier than the control mice weight. And that might be due to effects like something changed. Maybe their stomach is bigger, their satiety is different. It's not clear. It would be nice to have longer experiments with the overfed mice. And Jerry, does leptin have a saturating effect on controlling diet? Indeed. And we're going to talk about leptin's effect on diet in a second. Children and adolescents. Wait till the end of the lecture about children and adolescents. And yeah, the high fat part, high leptin, and we'll see what happens. So these are very good questions leading us to the math part. And we're going to do the math part now. So I'm going to switch gears. Do you like that expression? I'm going to switch gears. Let's switch gears and do a song. Have you ever wondered why our weight stays nearly constant? Give or take 5 kilos over decades, it's the same. Of course, there are exceptions where we really fluctuate, but overall it seems there's a set point for our weight. So if you want to know the answer, at least for rats, and you have a curious mind, let me take you by the tail and show you through the leptin circuit. I'll show you something that may help you understand. All right, so let's do it. I'm going to try to write the equations for these lines. And we started last time. We'll do it again so we can get used to it. Those of you who come from physics, math, etc., you'll see the equations are very simple. But please be patient for people who see for the first time differential equations. And just want to get everyone to the same level. And then the rest of the course, we're going to use math. More like a rapier than like a club. Okay, more like a toothpick than but always to try to make some points. In our case, I want to know what happens if I change some parameters like the fattiness of the food and the exercise. What's going to happen to the set point, so we can address the burning questions. How do I change my set point? This is a nice deep sigh of relief. And write down the equations. Okay, so I'm just contemplating where to do that. And the answer is in this area here. So we're going to start with the diet line. So we need to know how fat changes over time. How fat change, rate of fat change. And we're going to do that like this. We're going to get change from food. So when you eat, you gain fat, minus change from our from our metabolic rate, what's called basal metabolic rate, and exercise, and all our energy costs, where fat goes out of our fat cells and is used as fatty acids as fuel. So basically, in order to supply that fuel, fat mass reduces. And then there's going to be another change, which is the metabolic cost of fat itself. So fat itself has a metabolic cost. And we're going to separate that out so that we can make sense. So I'm going to write down the terms here. The change due to food is the food intake, grams per day, times a parameter that controls how much each gram of food is converted into fat. And that can depend on the kind of food you eat. So it's different for lettuce and for a cheeseburger. So alpha F here is rate of fat gain per gram of food. And its units are grams of fat divided by grams of food and time, per unit time. So that's a parameter. It's different. It has to do maybe with your intestinal system, your microbiome, your metabolic state, and most importantly, on what you eat. All right. Then we have our metabolic rate due to exercise and just the fact that our brain, liver, kidney, heart use energy just to keep us alive. And we're going to call that gamma E. So that's our metabolic rate. The rate of fat change due to metabolism and activity. And its units are grams of fat per unit time. So if I just to keep my metabolic rate going, 2,000 calories per day, etc., if I don't eat anything, my fat is going to reduce in that given day. Also my muscle, because some muscles are broken down to supply glucose to the brain, which only eats glucose, but most of our body eats the fatty acids fine. And the metabolic cost for fat tissue is, of course, the more fat you have, the more it is. And the parameter is gamma F, which has units of 1 over time. This has units of fat over time. So everything here has to have units of fat over time. Fat over time. Fat over time over gram food times gram food. And actually, I made a mistake here. This is gram food per day. Gram food per day, U. So alpha F is just gram fat per gram food, where it has the 1 over time. And gamma F is 1 over time times gram fat. So we have everything here in the same units. It's a great time for questions. Can you replace the black marker? Yes, I can. Question is, will it help? Well, I can see now that the black marker was ineffective. Alon, maybe you can see if we can grab a better black marker in the lab. Oh, there's this one here. Oh, it's a better one, thanks. Okay. So this is gram food divided by gram fat divided by gram food. Here we have grams fat per time, unit time, and here we have 1 over time. These are our parameters what we'd like to do actually is see, does this equation give us solutions like this? If we change food intake, how does food change over time? Does the rate of change of food give us the solution? That's what we want to know. That's how we work with these equations. You can simulate them on the computer, of course, and you will in your exercises do simple simulations to see if you put in some food and some food intake, how fat changes over time. It's kind of a lesson for life also. All right, so okay. So what I want to do with this equation is to solve it. So we're going to solve it in 2 ways. One is we're going to, first of all, ask what happens at steady state. Steady state means I give some food and I wait till fat doesn't change anymore. Like in this situation, or in this situation. Here there's more food, so the steady state is higher. Here there's less food, the steady state. Here there's even less food, the steady state is even lower. So what is the steady state? F steady state. What is steady state? In order to calculate it, I set the F dt to 0. Why? because there's no change anymore. That's the way, in all throughout this course, we're going to calculate steady state. It's saying things don't change anymore. So the equation is 0. Steady state, fat steady state. So if I set this to 0, then this gives me F steady state. F steady state is the fat that solves this equation. How I'm going to determine it? I'm going to move F steady state here. And then I'm going to divide by gamma F. Here, watch me divide by gamma F. And I have my first result. The more I eat, the more I'm fat at steady state. That makes sense, right? The slope, and basically this defines a line. F steady state is the amount of food I'm eating, assuming it's a constant amount of food. And the amount of food I'm eating. And this is the slope, this line. This is the diet line. If I give steady state amount of food, this gives me the diet line. This is the slope. And notice that the intersect at 0, how much food I need to eat. How much food I need to eat in order to have 0 fat, F steady state. How do I calculate that? I plug in F steady state equals 0. And I find alpha F over gamma F. And that's this amount. Gamma E over gamma F. And if I move gamma E over gamma F over here and divide by alpha F, I find that this U0 here is just gamma E over alpha F, which actually means that the more I exercise, more gamma E, this point moves over here. Exercise moves this whole line. And the new set point has higher eating and less fat, as we said last time. That's what happens when you exercise. So that's a cool result. Of course, I still didn't tell you what exercise does to the appetite null Maybe it does something to undermine that. Scary, but we'll deal with it soon. But if it doesn't, if it somehow magically doesn't do anything to the appetite line, we have a great result. That exercise makes us eat more, so yummy, but lose weight. Did you ever get this excited from an equation before? What else? If I eat more fatty food, so alpha F is the fattiness of the food. Remember, this is the food quality somehow. It's how many grams of fat I get for a gram of food. So if I eat worse food, more fat per gram, alpha F is larger. It makes the slope go up, and also makes the intersect go down. So it makes us fatter. Why does the intersect go down? Because the food now, we can eat less and still have fat, because the food is intrinsically fatter. See what I'm saying? So this is bad, but maybe it also changes the appetite line. We have to know what it does to the appetite line, right? So we have to stay tuned. Do you see how these equations are not just Greek letters? They relate to essential facts of life, for rats at least. Okay. But what about, so that gives you the steady state. I move from one steady state to another steady state when I change food intake. Yeah. What about this entire dynamics here in the middle? So in order to do that, we need to solve this differential equation. It's a linear first order differential equation. Its solution is found in textbooks in the lecture notes. Classic. It's I also mentioned it during the first lecture. It has to do, it has to do with a function that when you take its derivative, gives you minus gamma f times itself. This function is e to the minus gamma f time, because when you take this derivative, you get minus gamma f times e to the minus gamma f time, which is minus gamma f times f. See, that's the magic of this function, exponential decrease. And that exponential increase and decrease is going to give us these time-dependent shapes. So that's what we need to know about this linear differential equation. I'm just going to tell you the solution here. The solution of this equation is, okay, so I start at one steady state, F0, and I go towards another steady state, F steady state. And so the solution is going to have e to the minus gamma f time plus something. At time 0, this thing, let's look at it. At time infinity, this thing is 0. E to the minus number times infinity is e to the minus something very, very big. E to the minus something very big is 0, very small. So at time infinity, we're going to have f steady state. And at time 0, e to the minus gamma f times 0 is 1. So we're going to put here something, so f of t starts out at its initial condition. Because at time 0, this is 1, and f0 minus f steady state plus f steady state is just f0. This is the solution to this equation. And when you start at f0 and you put in numbers, you get an incredible match to the experiments. And you get that the time scale here is gamma f, time scale here, is 0.04 day to the minus 1. And the way to interpret that time scale, you can ask, when do you reach halfway? So if I have a process like that of weight gain, let's say, I go from some initial weight to some steady state and ask, how long does it take me to reach halfway? Of course, this is very important for when I diet. If I diet, I start losing weight, I want to know how long it's going to take me to reach halfway to the steady state. Very important, right? When I diet. So if we look at this equation, this halfway happens when e to the minus gamma ft is 1/2. Because then you have, this is basically the difference between f0 and f steady state. And this is 1/2, you made it to 1/2 the distance. And that means that, I'm going kind of fast here. You can find all of this in the lecture notes. E to the minus gamma f t 1/2 is 1/2. And the response time, the half-life, and this is going to be for in all of our course, when we have an equation that has some rate constant like that, the half-life is always given by 1 over this rate constant. This half-life, in this case, is something like 20 days. So for these rats, it takes them 20 days, a couple of weeks, let's say, to get halfway from the beginning to the end of their weight gain, given constant amount of food. Right, so the bottom line is, this equation not only gives us a linear diet line, which looks like the data, it gives us the entire trajectory. And from this, you can calculate what these parameters are for the particular food that these rats were eating. And if you want to calculate alpha for a new food, you can do this experiment and get how things change. And you basically experimentally can test your model. And I'm going to let you digest this. And the way I want to do that is to do a breakout room. This breakout room is your opportunity to meet other people. Maybe some of them have a computational background, maybe other have biological background, and talk to each other about what we've just done before we go to the appetite line. Okay, so welcome back. So now it's a great time to ask questions by chat if there's still unresolved questions. I know this is fast for some people, and you'll have your lecture notes, and you have each other. So we use the heterogeneity of this class. If you can find people from computational background or more computational background, you can help them, be helped by them, help them understand the biology better, maybe. I know some people weren't assigned to a room. I apologize for that. Okay, so any questions? So this is the time to chat. Please, I want to wait until there's one question by chat or comment. The comment could be, that was too fast, or that was just right for my speed, or I have faith in you, or I heard one question was, is this similar to other hormone systems? So other hormone systems we'll see are more, they have additional principles. They're quite amazing to do more things in the system. But the basic equations, this is like a building block that's found also in other hormone systems. Okay, I'm gonna, so we have the diet line, steady state, and half-life. All right, so we did basically the diet line. Let's take a nice deep sigh of relief. Superb. We have our parameters. I'm going to write down for, on this graph, we have alpha F, how food is converted to fat, how fat is removed. That's gamma E and gamma F. And so those are the parameters that govern this part of the feedback loop. Now we want to go to the appetite line that has to go through leptin. That's our next challenge. So. Great. And the idea will be to see how leptin is made and how leptin shuts down appetite. So starting with how leptin shuts down appetite. Leptin inhibits food intake, or more precisely, food-seeking behavior. Right? So in a free eating situation, we're going to write down that the food intake we have is going to be a function of leptin. Now, leptin is a molecule. It's a polypeptide. It's a little piece of protein floating around in our blood. And the way it does it work on cells, like neurons, is by binding the leptin receptor. And this has a chemical binding-unbinding, which has a typical affinity called KL. KL is the effectiveness of each leptin molecule in binding the receptor. It's basically you can define it experimentally as the concentration of leptin in the blood needed for 50% inhibition of appetite. Right? So let's plot that out. This is leptin concentration. And this is inhibition of appetite. So if I have no leptin, I eat until my stomach gets distended and I'm nauseated and I can't eat anymore. That's U max. Okay, so U over U max here is 1. And the more leptin I have, the less I eat. And the 50% point is reached at this KL. That's the leptin. And leptin resistance means higher KL, and therefore less effective leptin reduction of appetite. This is leptin resistance. Okay, so we need to give this function a specific form. And I want to say that a nice thing about physiology is that these functions almost always, when you experimentally measure them, have a certain form. And this certain form comes from the physics of a molecule binding a receptor. And so I can safely write down what this form is. U over U max, 1, leptin, KL. And this form, the name of it is the Hill equation. And the Hill equation is familiar to everybody who studied biochemistry, like a hemoglobin binding of oxygen, or etc. And it's a good description also of hormones in the blood. And if you want to know the physics, the biophysical solution to it, you can find it in the lecture notes in a simplified way, where n molecules of leptin bind a receptor in order to, and when the receptor is bound, it does something inside the cell. And so the Hill equation has two parameters. One is KL, and the other one is what's called the Hill coefficient, which controls how steep this function is. It always goes to 50% at KL, and the bigger n, so this is n equals 2, this is n equals 4. The bigger n, the more steep it is. That's called cooperativity, and we can be pretty sure that leptin follows a function like that. We just don't know what n it is right now. Did I explain myself? Let's take a nice deep sigh of relief. Please chat any questions. Can you write bigger? Thank you. U max. Hill equation. All right. So we can say that U is U max. And what is this Hill equation? I didn't write it down mathematically. It's very simple. It's L over KL to the power n. So this equation, you see, when L is 0, you get U equals U max. When L is KL, this is 1. So 1 to whatever power is still 1. So it's U max divided by 2. It's halfway. L equals KL. And when L goes to infinity, this goes to 0. So this function is a good description, by physical description, of hormone receptor binding. And now we just need to, so here we have U max and N and KL, these parameters. We just need to know how fat produces leptin. Okay? That's the only thing that remains. And so I'll write down the equation, and then we're just going to just plot this null line. So the way that what we can determine from this graph here is that the rate of change of leptin is fat, but it also requires food. So if there's 0 food, there's no production. So I'm going to write U times F. If there's 0 food, there's no production. And we put some parameter, which is the rate of production of leptin per unit food intake per fat. And leptin is also degraded. It's removed by the kidney. This is the removal in the kidney. By the way, the half-life of leptin in the blood, log 2 over gamma L, it's the same equation, is 40 minutes. So leptin is relatively quickly removed from our circulation by going out of the kidney into the urine. So we know these parameters, and we can, because it's 40 minutes, it's so fast compared to everything else, we can say that leptin is at steady state. I'm just showing the same steps as before, leptin steady state, and we get an equation that leptin is food times fat, and times some parameters, alpha L divided by gamma L. And I can plug this into here. So now I have U equals intake times fat times some parameters. And this gives me the appetite null line. because for every level of fat, I can now calculate the appetite. And I'm done. Take a nice deep sigh of relief. And when I look at this experiment here, we can erase all this intermediate steps and write down the answer, which is approximately the diet. We have the diet line, which it has its own equation, and we have the appetite line, which goes like alpha F KL divided by gamma E, U max divided by U minus 1. And this gives you this line, which goes to 0 here at U max. You see, U max, when U is U max, you have 1 minus 1 is 0. And when U is very small, you have 1 over U, so you have this increase here. And you get a really nice fit to the data when n equals 2. So we know the Hill coefficient. And that's it. There's no other parameters here. And then we have our diet line, which, as you recall, is alpha FU minus gamma E divided by gamma F. So we have equations for these two lines, and we're golden. We have everything we need to know about weight. And now we can answer some. So we have appetite equation, Hill equation, leptin equation, take a nice deep sigh of relief. And now we can ask about dieting, right? So how can I tell you I lost weight and I finally fit those jeans? Yes, I know, I'll keep those brown sacks. Pretty soon I'll gain those pounds back. Our film expert here is laughing too with me. It's a cycle that never really ends. All right, so we're now in the position to answer the burning questions at the beginning of this lecture. What happens if I eat food that's more fatty in the sense that it gets converted to fat easier? What happens if I exercise? And you notice that things like alpha F, which is the food quality, appear in both null lines. All right. Here you have f squared, so this will be like a square root dependence. F squared. You do square root to get rid of the f squared, and maybe I'll write it like this: 1 over n. So if it's f squared, it's square root of what's here. So the quality of food appears in both. The exercise, gamma E, appears in both. KL, on the other hand, and U max appear only in the appetite null line. So the satiety, how much maximum food you can eat before it's repulsive, and KL, your leptin resistance, appear only in the appetite line, not in the diet line. So now we're in a position to say, suppose we change one of those parameters, what's going to happen to our set point? So we draw out our null lines, which look like the model null line looks like this, and the diet line looks like this. And now we can go parameter by parameter and look at our set point. And that's what happens if we do, let's say, a 50% change in, let's start with easy things, leptin resistance. It only moves this null line. If I increase leptin resistance, I get fatter. So this is what happens when you change KL, leptin resistance. What happens when I change U max? As you say, somehow, maybe I eat so much that my stomach gets bigger or something like that. I get an even bigger difference. And by the way, this difference means I eat more and I get fatter. Or maybe, but another way, if I can reduce my stomach, I'm going to eat less and be thinner. hope I'm explaining myself. It's burning issues. And now what happens when we change parameters like exercise? So exercise or basal metabolic rate controls this gamma E. So as I said before, if I exercise, I shift the blue null line like that. But on the other hand, I shift the F null line like that. So I get a situation like this. And you need to do the exact calculation to see what it does. It turns out that exercise it's still dominated by the diet line. And you eat more and you lose weight. That corresponds to what actually happens to rats. Giving them a wheel, they eat more, they lose weight, but they lose relative weight more than the relative gain in eating because of the slope of this thing. So let's say 30% weight, if they give them a wheel, 30% loss of fat and 20% gain in eating. That's qualitatively agrees with what the predictions of this model. So you can now write down a lot of experiments to do. Give them a wheel and change their Umax and give them hyperthyroidism, whatever. No. See what happens. What about food fattiness? So I'm going to change alpha F. I'm going to make the food fatter, or more precisely, increase the rate at which each gram of food translates to a gram of fat. Increase that parameter. And you see that appears in both the appetite line and the diet line. And what happens is this. So there's a lot of weight gain. Alpha F, weight is very sensitive to alpha F. And almost no change in eating, slightly less eating grams. If you eat more calories because the food is more caloric, maybe, but there's almost no change in the amount of food you eat. And that appears to be true for rats on what's called high-fat diet. That's experimentally used to test obesity, etc. And so we can draw this kind of the diagram to predict what's going to happen with different interventions and answer the burning questions of parameter changes. Okay, so there's some questions here. Where is leptin produced? Is it just all the time roaming in the blood? So leptin, thanks for the question, leptin is produced by fat cells. In fact, until 1994 or maybe 50s, where it was suspected that there's this hormone, in 1994, where it was actually nailed down molecularly by Jeffrey Friedman at Rockefeller University, fat was thought to be a boring container for storing extra fuel, which we carry around. After 1994, the view of fat has changed into a smart organ that produces different kind of hormones that affect both your brain appetite, your immune system. They're called adipokines. So fat is upgraded to a smarter organ. It's produced in the fat cells. The hormones are always produced by a sender cell, the endocrine cell, and move in the blood to distant organs, in this case, from fat to brain. Hope I answered your question. Next lecture, we'll study insulin produced by cells in your pancreas called beta cells and talking to your liver, your muscle, your fat, etc. So these are hormones. Endocrinology, the study of molecules produced by one organ to signal to other organs. Yeah. I'll be trying to studying the transfer function of this system. Transfer function is used in engineering to take a black box, for example, give it some inputs like sinusoidal inputs, measure the outputs, and you can get a very good mathematical model of what's going on in the system by these kind of measurements. It's called the transfer function method. And that's a great idea. And I think it would be quite revolutionary to design an experiment like that and get the transfer function, the linear response, basically these arrows from an experimental system, and even possibly from human beings, because if you ask people to make a small change to their diet, that might be ethical. And maybe that will be a groundbreaking way to study. So thanks for that question. We have a new project now. Talk to me if you're interested. So you're saying the KL, or apparent KL, this halfway point of leptin effect or leptin resistance, is at the similar scale, both the molecular level, leptin binding to the receptor, and the physiological level, controlling diet. There are no other factors coming into play. So this question is a detailed question. So I did say that KL describes the biochemical interactions between the leptin and its receptor, whereas in fact there's other steps in here, inside the neuron cell, etc., that lead to the physiological effect, which is inhibition of food. And they're not fully understood, of course, because what the brain does, food eating behavior, is far from understood. I mean, we know the next step from leptin binds to two kinds of neurons, POMC and peptide Y producing, I think. And it inhibits hunger producing cells and activates satiety producing cells. So it has opposite. So there's some part of that circuit is characterized in the receptor. But then what goes on beyond that in the deep brain? What does eating behavior mean? That's like cutting edge research, etc. So what I'd like you to remember in this lecture is this KL is the physiological answer. It's the amount of leptin needed for 50% effect. And it could be different from the amount of leptin needed for 50% binding. So that's two different things. And I hope I was clear, and thanks for this clarifying question. Is fasting good for leptin release and control? Fasting inhibits it. Fasting stops production of leptin so that you actually have more appetite, at least in the beginning. So less leptin. So that's an evolutionary part of the circuit, as I said before. Maybe that if you're fasting, the body would like you to have food-seeking behavior beyond what's appropriate for your normal level of fat, because it's food times fat. I tried to ask before about persistent changes, meaning that one-time change that will affect the diet and appetite lines. So Itai, I didn't understand. Could you rephrase your question? How do you explain in this model high-fat diet in which people lose weight, like paleo diet or ketogenic diet? So the question is, in human beings, if you sometimes you eat high fat without carbs, that's the point, you can lose fat. In my opinion, one reason for that is that it's the carbohydrates that make this little arrow here. It's the insulin. And if you don't eat carbohydrates, it could be that that the circuit is different than when you do eat carbohydrates. So I think that's the important aspect in those ketogenic diets, which, by the way, are effective but have side effects. So be very careful when you think about them. Especially some of them are just fads and are fake science, but others are like ketogenic diet are studied and recommended in certain situations, but not all situations. Given choice of different kind of food, would you the cycle control this choice as well? So I also want to say that this diet model is for rats, right? And in humans, there's a lot of psychological factors associated, let's say, with the kind of food you eat and cultural factors. So extrapolating to humans, it should be done with a grain of salt. So notice that I have a lot of these puns. I don't intend them, but they just come out in this lecture. So that's kind of a fun thing. All right, so let's bring this puppy home, okay? So there's two more fascinating topics that I want to discuss with you. Now that we have this model, this maybe should be proud that we have a hypothesis from which we can predict many things and test. And of course, the idea is that experiments are always going to be different, that we can improve the model, etc. But at least it's a starting point to understand where the weight point is and why is it that we have the weight set point, what happens to it in different situations. Leptin resistance shifts this null Klein and is in charge of a lot of obesity in the world, a growing problem. And all right, so I want to ask how, now I'm going to move to humans, and I'm going to tentatively use this model, despite what I said before, to make a kind of conclusion, a tentative conclusion about why is it that human beings have such a different weight set point? What parameter primarily changes between human beings to make us so different in our BMI, body mass index, mass divided by height squared, or percent fat, or whatever. So, of course, your height makes a big point. But if we factor that out, percent fat, there's a wide variation, let's say factor 4 between people. What is going on? What parameter is changing? So I want to address that from the point of view of leptin by telling you about many different scientific papers that ask what's the relation between leptin level and percent fat. So you take different people, different populations. What's the relation between leptin level? So here I'm going to write the percent fat. And in different people, you can go from 10%, kids a lot of times, 20%, 30%, 40%. That's really obese, okay? Or BMI, body mass index, where we're above 30, we're talking about obesity. Above 25, we're talking about overweight. And here I'm going to pull out leptin in concentration, nanograms per milliliter or things like that. Okay, so it turns out, I'm going to plot it here in log leptin even, or no, just leptin. It rises, fatter, more leptin, and is a good approximation that leptin goes like fat squared. So if this is 1 unit, it's 40%, we're talking about 60. There is variation around this line, but it's less than the variation along this line, well, much less. So that's the data from different studies, different genetic backgrounds of people, kids, male, female, and there's difference between male and female in the lines, but both have L goes like F squared. So what's going on here? So when you use this model and ask which parameter here, which parameter can change between people to give leptin goes like fat squared. Not all parameters do that because it depends how the set point is changing. So if this was exercise, let's say these are exercises, non-exercisers. Exercise shifts this in the wrong way. You have more fat, let's say less fat, but more eating. What we want, if leptin goes, as I told you before, leptin production goes like food times fat. So in order to get F squared, we need food to go up and fat to go up together. We need a parameter that makes intake and fat go up together. So it can't be quality of food or exercise. It has to be one of these. And when you calculate, you get to the conclusion that this variation is most likely due to Umax, which is the amount of food I can eat until I can't eat anymore. And that's an interesting thing. There's also leptin resistance. So differently, people with a lot of fat are leptin resistant, but it looks like a secondary compared to the Umax. And that means that one way to go down here is things that affect Umax. Umax, for example, gastric bypass surgery that restricts your stomach and changes where your food is digested is a very extreme procedure, but it does get weight loss, at least for a while. And it affects Umax, probably other parameters too, but that's one way. Or drugs that affect your satiety, like GLP-1, which is a hormone produced naturally in the gut when you eat. And it makes satiety, tells the brain of eaten, thank you very much. It also tells the brain, beta cells to make more insulin and things like that. So GLP-1 or things that reduce Umax are likely to reduce leptin and be one way to think about weight loss. But this is all cutting edge research, etc. So the variation here can be food quality, could be exercise and things like that in the perpendicular direction. So that's about difference between people. And now the topic of aging. So Many of you are in your 30s, 20s, 30s, 20 something. Maybe your weight has been constant for a long time. Maybe it looks like life is going to be infinitely like that. So let's examine that. How do these parameters change with age? Yeah. So the parameter that changes with age the most is the basal metabolic rate, and that's part of gamma E. What is basal metabolic rate? It's the calories per day needed to keep you functioning. When you're not moving, you're lying in bed, let's say, watching Netflix, your brain is still, let's say, functioning. That's 20% of metabolic rate. Your liver is functioning, 30% of metabolic rate. It goes liver, 30%, brain, 20%. Then there's kidneys, yeah. Then there's the muscles for your digestion and your fat and your heart, yeah. And there's more stuff in your body just to keep the ion concentration inside your cell and outside your cell different. There are pumps working, important part of basal metabolic rate. That part, you can say per unit mass or per unit surface area, a lot of times it's computed, changes with age. How does it change with age? And of course, when it changes with age, let's say if the metabolic rate decreases, it's the opposite of this arrow. You're going to eat less and get fatter. That's why you should care. Actually, you shouldn't care. You should learn to love yourself. I don't know what you should, but this is not a course on ethics or mindfulness. It's like a nice deep sigh of relief. That's mindfulness. This is age. This is your age, and this is your basal metabolic rate, abbreviated BMR, and different ways to measure it. And I'll get into it. So I'm going to plot here age 20, age 30, age 40, age 50, and beyond. So it turns out that until age 20, basal metabolic rate does this. So kids, babies have higher basal metabolic rate. Energy cost per unit mass or per unit surface area, that's declines. And when you think about it, kids are really skinny. So I just took my daughter, my youngest daughter, Carmel. I just had her 8th birthday to the doctor, and she measured her, and she has body mass index 14. I have body mass index 25. That's on the border of overweight, and above 30 is obese. Of course, the amount obesity for children is different BMI than obesity for adults, but she's much thinner than I am, percent mass and stuff. She might have 10% body mass, yeah. And this changes. And then it hits a plateau. So for 3 decades, your body mass is not changing, your basal metabolic rate is not changing for most people. And that is beautiful because forever you're going to be young and thin forever. And then at age 50, for most people, it declines, which means that every gram you eat sticks to you more. So what can I do if I can't diet? And we still don't have a GLP-1 weight loss pill. Exercise to kind of reduce gamma E or increase gamma E, right? And eat good food that's going to reduce alpha F. And maybe walk the Buddhist path, accept your set point, and learn to love yourself. All right, see you next week where we change to a new system, the insulin glucose system. Thank you very much for your questions. Those of you that want to talk to me with your camera on, please stay tuned and open up your camera. And for the rest of you, goodbye.